When Phil asked me to come to this meeting, I complained that I had already agreed to be in Stockholm, and so he arranged for me to give the closing talk because he figured I would just make it in at the airport in time. But he was so persistent, which is what I love about him, that I decided to do Stockholm virtually. So I've been up most nights doing Stockholm, which is off the time scale that we're on, so I'm a little tired. So what I thought I'd do, if I can find what I did with the pointer. Someone know what happened to the pointer? Did I set it over there?
Did you put it on the... Did you put it...
No. Is there one in there? It'll show up. Is take you along a path that we've been going toward integrating distributed multiscale information, generally with the objective of creating what we like to call visible virtual cells. So this involves a lot of different microscopies to build these kind of cellular constructs. And then the integration of information such as structures determined by X-ray crystallography into those constructs. So these are the kinds of things that come from microscopy. At least for those of us who are interested in the nervous system or tissues, this would be a structure, a gap junction from genus Acinski, a mitochondria that would fit into this little space and a synapse. Those are synaptic vesicles. You saw a lot of images of those. This synapse would be at the end of this dendritic spine. This branch of a dendrite would be a little piece on this big green cell. And of course, that fits into the layer of green stuff that you see in the slice of the brain, et cetera, on up to a non-invasive image of a human. I promise you it's non-invasive. That's my wife. She's still with us. But really, the mesoscale that you heard about, that Graham mentioned, is what we consider to be this region. And we think we sort of coined that term. We're kind of excited. We consider it, we keeps expanding. It goes from a nanometer to a millimeter in our world right now. But a lot of people have been involved in this with us, and I'll go through. Usually you put these credits at the end. I'm putting them here because if I forget anybody, I like to have you come up afterwards and I add you to the list. But this is a lot of people who've been involved in this effort around here for 25 years. And as Steve Peltier mentioned, we struggle with the problem of being kind of the biggest data producers around. As you'll see, some of the data that we take to try and do visible virtual cells are large at every level. So let me give you a sense of what I mean by large. If you wanted to characterize a whole cell, and this is a bad cartoon in terms of what it actually represents, and you wanted to do that across something like an entire mouse brain, you'd really struggle. A mouse brain, let's say, is a centimeter on a side. Okay. If you wanted to do that at the level where you could see microtubules, what would your resolution need to be? It'd need to be about 10 nanometers isotropic resolution because a microtubule is about 24 nanometers across. If you wanted to be able to see, just based on their structure, every macromolecule, you'd want to be closer to 1 nanometer or 2 nanometers. You wouldn't see the crystal structure, but you'd see the envelope of things, which you've seen in most of these depictions of molecules anyway. So how much data would that be for one whole mouse brain, one mouse, one moment in time? It's a zeta voxel. Okay. So you already heard that there's not enough iPhone memory. Well, that's not quite true. You could do this if everybody had a little piece of this. But let's be reasonable. If you wanted to do a whole brain or any organ, you could break it down to how many cell types are there. Let's say in the liver, 5 major cell types. In the brain, maybe there are 2,500 major cell types. And if you had a formula for how you create those visible virtual cells, and you had a lot of people doing the work, and you had a way to bring it back into a common scaffold, a brain atlas, you could probably do it in 20 years if everybody agreed to do it in a way that was rigorous enough to integrate that information. So that's kind of what we're pushing, is how do you come up with a way to harvest that data in a consistent way, different people doing different component work and bringing it together in sort of a Google brain. So at the National Center for Microscopy, we sort of work on this by building the instruments, by coming up with workflows, data flows, utilizing high-performance resources that other people generally pay for, and putting those data in databases like the Cell Center database, the Cell Image Library. You can go to the Cell Image Library and look through the way this is represented. And it involves workflows that use tools from the community, now more or less operated as services, to image, reconstruct, segment, mesh, analyze, and then in many cases simulate, as you've been hearing. We even went as far with a philanthropic gift to create a Google Brain. We call it the Whole Brain Catalog, and there are a lot of apps to play with it using an open source game engine. Well, what do we consider to be the challenges? Electron microscopy works all the way down to an angstrom, actually sub-angstrom for material science, but for biology practically, we're hovering around a nanometer. It covers a wide range because you can turn an electron microscope into kind of an aerial photography machine and go out and do tens to hundreds of microns across a field in 3D. It's just a lot of data and a lot of work. Light microscopy in recent years has become a super resolution technique and starts to penetrate in this domain if you're looking at fluorescent molecules. But of course, it's always been capable of working across large areas. And you saw that very well demonstrated by Marcel's talk, looking at whole portions of tissue and the way they organize in the whisker barrel cortex. We do this with systems like looking at a tumor in its microenvironment, mouse models that express tumors in the optic nerve. This is the optic chiasm. The eyeballs would be here. And you take data with the whole diversity of techniques. This is a spread retina preparation from mosaicing light microscopy to count how many cells are still there. This is a technique I'll show you, Steve showed you, of serial block face imaging. And these come together. So light microscopy, labeling each cell type, making a map of that's the optic chiasm. You go down quite a ways, you get to both eyeballs. And then you can map cells, the mitochondria, their shapes, their quantitative characteristics, and so on. This all goes together and then provides a scaffold, if you like, for people who work on this disease, and this goes in a catalog. So the general things that you do are image capture, terabytes, segmentation, usually gigabytes, display and analysis, megabytes. So it's big data all the way through. Light microscopy runs out of resolution when you're trying to deal with the details inside of cells, and you only see what's lit up. You only see what you've been able to attach fluorescent proteins to. If there are 30,000 proteins alone, you're going to be at it a long time trying to put in markers for each molecule. Electron microscopy has an advantage in resolution. There are some really special electron microscopes. Here's a person for size reference. So she's loading a sample. This is a one-of-a-kind 3 million volt electron microscope in Osaka. We made it capable of doing electron tomography, so it's now most often used for biology there. So this is that same sort of spiny dendrite that you saw. These are the little nubs that, at least in the cortex, 90% of the connections, the synapses, are made on. This just happens to be from a mouse that expresses a form of mental retardation, Fragile X syndrome, and the spines get a little tortured. But you can do this, and you can't do this everywhere because it's a one-of-a-kind microscope. But there are new tools, like the one that Steve Peltier showed you, that are coming along. This is a scanning electron microscope. One could probably make a system like this for $20,000 with a tiny little microtome and a tiny little column. that would generate data sets like this for every laboratory in the world, kind of like a light microscope. It's not beyond current technology, it's just not the way the technology is delivered. So you see we're going through lots of cells here. There's the nucleus. This is downsampled by 15x just to show you this data set. You can take a 32 by 32K scan, mosaic 3 by 3. I can do this with a slightly different machine with an image every 2 seconds, so 10 billion pixels every 2 seconds, to make maps of cells very quickly. Okay. We can, just like in light microscopy, paint proteins genetically now for this kind of a technique. So this is PSD95, one of the synaptic proteins that Terry talked about. So you can go through and mark these things and have them come out. We'll talk about that a little more in a moment. So how are these tools being used? These are becoming very popular. As you heard from Marcel, a lot of people are interested in the wiring diagram. Here's one project from George Spyru, who comes to San Diego to use our technologies from West Virginia. He looks at these big, large field areas. This one actually isn't so large. It's only an 8K wide field in pixels. But each one of those cells was to be mapped like a whole virtual visible cell. How does he do that? The hard way. He's in an undergraduate institution. There are a lot of kids who want recommendations for medical school. Every one of those numbered cells is a new letter of recommendation for medical school. Okay. So it's not scalable, but it's practical if you're trying to get a paper out in a couple of years and you have a large student body to do this sort of thing. So here you see a movie. Again, this is downsampled by more than 10x just to show it. Now, as long as we're not broadcasting this presentation, you can have the soundtrack, but I think the copyright or whatever on this is not legally. So here's the nucleus, here's the cytoplasm, there's the Golgi apparatus, those are all mitochondria. This is just showing you a little piece of this reconstruction. This is one plane in a couple of thousand planes. Again, downsampled, otherwise you'd be able to see microtubules and filaments, but you can see all of the synaptic vesicles quite clearly. What's this project about? Well, briefly, it's about looking at the development of this part of the nervous system, which has to do with auditory processing. This animal is from postnatal day 2. Postnatal day 2, there's a presynaptic terminal, which you see rising out of the complicated neuropil. It's trying to own the neuron. It's got competitors. The neuron has 2 other suitors. By day 4, one of them has to win. How does that happen? So they're mapping what molecules, what adhesion molecules, what cadherins are involved. So this would be what you'd see as a growth cone. In tissue culture, that's the axon coming down from a neuron that's far away. And you'll see who it's trying to seduce. Here's the nucleus, there's the cell. That's one letter of recommendation. There's 100 of these now. And they can be mined for other things. Here, early in development, this is before these 3 colored cells have figured out which one of them is going to actually get to put its pod down. As soon as this paper is published, these will be available as visible cells for virtual use. People can annotate other features other than the ones that were used in this study to then have their way with their simulations. Here's a little later. I won't go through all of this. Eventually, one of these guys wins. The electrophysiology is charted. The other ones die away. They're trying to understand how that works very quickly in early development. Well, if you don't have a whole herd of undergraduates trying to get into medical school, then you need other ways. And Steve talked about crowdsourcing. The first thing we do before crowdsourcing is we work with smart algorithms with Tolga Tazin's group, Liz Juris at SKI at Utah, to come up with ways of automatically segmenting the plasma membranes Other tools are used in the workflow to then give you the mitochondria or the synaptic vesicles. You train this on a small data set, and then you send it to the cloud. Okay, we use the NSF resources basically as our source of computing for this sort of thing after we've trained our algorithms. There are probably 20 major labs working on different segmentation codes, automatic methods. So our strategy is basically to get as many of them as possible, to open those architectures, to provide those tools, to give them a way to build them so that they can drop into standard workflows, just like you do in bioinformatics, so that you can mix and match and take care of which ones were used for every one of these that goes up on the sort of visible cell site that we're creating. Again, if you want to make life easier, you can selectively stain the cells, which you can do genetically, as I'll show you in a moment, but this is the way Golgi and Cajal got the Nobel Prize over 100 years ago by a special metal staining, which turns out to be just perfect. What was good for light microscopy and Nobel Prizes some time ago is at least good for electron microscopy today, but probably no Nobel Prizes. But here you see those cells as they come off of this slicer, this microtome inside of a scanning EM. And of course, now that you've got them there, you can pick one of them, seed it, and have it stand out from the neighbors, as you'll see in a minute. So this gives you the envelope of a complicated cell. What about what's in it? Well, before we go there, let me point out that there are other cells. The nervous system is a jungle of cells. Here's probably the most important cell in the nervous system. This is an astrocyte. These tiny little processes have been hidden because they're too small to see in the light microscope. But with this technique, we can follow them in detail. And then you can ask, if you have a way of painting just the mitochondria in a single cell, what do the mitochondria in a single cell look like? Or here, because we made the lysosomes darker, you can pick those out. Now, I thresholded this just so those more dense components would come out. But if I played with the lookup table here, I could show you all the rest of the ultrastructure here. And someone can come along with this data set and say, oh, I want to see the synapses in the vicinity of this and know if any mitochondria are near them. So that's a way to remine this data after it's published for its first finding. Here's a data set we took in San Diego for friends in Helsinki, who then bought one of these microscopes, but we showed them how to do the staining. This is delineating in mitosis the ER as stained, and then hand-picking all of the chromosomes. And then figuring out what happens to the ER. So this is just ER associated with the mitotic apparatus, which people always talk about microtubules. They're not even thinking about the ER, which forms an enormous network around the mitotic apparatus. Then you can fill in all of the mitochondria, all of the lysosomes, and look how crowded the cell becomes when you actually automatically segment the rest of the ER. That's just endoplasmic reticulum in a mitotic cell. How many of you knew to think that that space was so crowded with membrane tubules? So now, Graham, you have to pack that with everything else. So to make your life even more interesting, here's a microscope we built. Old microscope, new camera. Camera was $4 million in parts. Microscope I salvaged. It's a 400 kV microscope. The field of view was larger than a millimeter. So this image, each one of those little tiles is an 8K acquisition from this mega camera. So it's 640 billion pixels for one image before we do the 3D work. So these machines are becoming more available. When we built this, this was kind of extreme construction 7 years ago. Now the parts are cheaper. You can do it in different ways, but this is where this community is going for virtual visible cells. Here's an example of actually recording a cell at that resolution. It's a single cell, and it's one that was of interest to this student, Florin Vercerno and Anna Lizakowski at the University of Chicago. They wanted to have a cell reconstructed where the cell actually rocks. Here's what I mean. This is a cell, when I move my head in one direction, the top moves, and the electrical activity increases and more transmitters released. I move in the other direction, and it decreases the output. So the top is flexible and the bottom is stiff. They don't know how that happens, and it changes the output of the cell. So we said, well, what's in there, and how would you actually give it mechanical properties and put it in a mechanical model? So what Florin did is he took sections every half micron, did tomography with that big machine that I showed you, stacked them up, and again, this is downsampled for the movie, but he described then this striated body that's kind of like a Chinese finger puzzle at the waist of the cell that gives it its stiffness, and then he was also able to describe all these cables of actin filaments and how they associate. Now, those of you who are thinking about metabolism might also notice that there's this little herd of mitochondria that you see all up here. So whatever's going on in this cell, we presume that most of the energy's got to be up here, but the transmitter release happens at the other end, and that's energy requiring, so there's all kinds of modeling to do on this. So now the paper's published on this just this year in PNAS. So the model is available and people can pick it up and do whatever they want with it. Switch these surfaces to finite elements, do the mechanical modeling, and so on. Here's an example from one that Terry and Tom didn't have time to give, but it goes back to the first use of this kind of tomographic data for tomography. And it was kind of a fun project. I think Tom has recovered from it. So it was a project that started more than 10 years ago with Rick Shoup, a graduate student in Darwin Berg's lab here, looking at something called ciliary ganglion neurons and their types of acetylcholine receptors. So we knew from his thesis work that there were multiple types of receptors and that they formed these little patches on the surface of this spherical cell. And the patches looked a little bit like this, and we could tell these two types of receptors for which there was different physiology were a little bit asymmetrically distributed. From the high-resolution reconstructions and the physiology, we could make a map. And so this is from serial section tomography, just like that whole cell, except it's this little patch on the surface of a big spherical cell. So these are like the little strands on a shag carpet. They're like a dendritic spine, but they lay in a little crevice, kind of like a divot on a golf course. And the presynaptic terminal, which has been peeled off, is where the transmitter's being spit. And Graham and Terry both talked about that. So what we did is we painted and decorated this with the information that was appropriate for the specific hypothesis that we decided to test. Many other hypotheses could be tested, but we wanted a science paper, so we picked what we thought was the coolest one. Okay. Which was a controversy that existed in the literature about whether or not in the nervous system, synapses spit directly or out to the side. I'm saying this in a rather peculiar way, Tom, forgive me. So the dogma in the nervous system is that if there's a synapse, that's where the signal occurs. A lot of us thought for a while that transmitter could be released away from a synapse and diffused to receptors. So it was kind of the loose in the juice versus the direct hypothesis. Nobody liked loose in the juice because it added complexity to thinking about the wiring diagram of the nervous system. This study proved that loose in the juice is the only way it works. By a simulation, okay, because the only way that we could replicate the physiology at the time was to show that a certain class of receptors were activated in the M-cell program, which Terry showed an exquisite example of yesterday. Now, this is one of those examples, and Tom can give you the reference later, where a year after the publication, we pissed off people enough that somebody actually did the physiological experiment that proved that we were right. So again, this is this virtuous circle where simulation on top of real data causes experiments to be done, and now we can go to the next. You can see there's all of the people involved. Okay. So again, it even got one of these write-ups. Again, this is almost 10 years ago, I guess, of what the big thing was. Now, There's a problem. If you really want to do sort of painting and decorating of where everything is, you can do some of it by just recognizing structures. So here's an actin filament in a synapse. There's the synaptic density. Actin works out pretty well, but if you wanted to see things like Terry talked about, cam kinase, you'd have to have really good eyes and imagination to know that those things are cam kinase. Probably. Have I convinced you? They're the right size, they're in the right place. How can we make them say, hey, guys, I'm cam kinase, kind of like you do with fluorescence microscopy, but it doesn't have the resolution. So we took on, more than 10 years ago, the task of trying to come up with something that was as smart as GFP, but for EM. This is Roger Chen's lab and my lab. So that we could do this sort of painting and decorated in visible virtual cells, sort of molecule by molecule, actually multiples at a time now, to paint in around hypotheses what it is that we want to understand the location of. So the first one that we published was about 10 years ago. It's a tetracysteine system, Flash and Riash, but just within the last year and a half, 2 new molecules have appeared that we think are better. One is called the mini singlet oxygen generator, or MiniSOG, not based on the jellyfish protein at all, but based on phototrophin 2 and the light oxygen voltage domain. It's 106 amino acids. We've attached it to at least 50 proteins now for correlated light and EM. Just last month, or a month and a half ago now, I guess, with Alice Ting, a former postdoc in Roger's lab, we published another one. This one is not generating singlet oxygen to do the painting. This one is an enzyme. So it's a small peroxidase from P, and there's no disulfide bonds, so it tolerates operating in the cytoplasm. Actually, we haven't found an environment it doesn't work in. It runs after full glutaraldehyde fixation, and it runs at 4 degrees. So here's one of these. Again, mitochondria is the example. In a living cell, this is the MiniSOG. After fixation and creating a light opaque object by depositing diaminobenzidine, and then after osmium treatment to make the whole thing black and cutting a section, you see the mitochondria in the EM. Now, we've already done this with a whole variety of specific mitochondrial proteins looking at signaling pathways, some of the stuff that Susan's lab and our lab are doing. So we can dissect signaling mats a molecule at a time in the context of these kinds of hypotheses. Here's another example. That was the light microscopy of a cell living. This is with alpha-synuclein, which is one of the proteins that's overexpressed in a majority of Parkinson's disease cases. And here, this presynaptic terminal is mapped for that. Now, if you look over here, you'll see some coated vesicles, clathrin-coated vesicles. Well, maybe it'll come up in a minute. So this is a non-expressing cell, and we asked, there's one, what kind of resolution are we getting out of this kind of super resolution tomography, which I don't have time to explain to you. It's as many as 2,000 images of the same object in order to give us molecular resolution. So we asked that of that exact same sample by saying, well, let's look at one of these otherwise unstained, just lightly stained coated vesicles. Can we see the triskelions? Can we see the clathrin baskets? And indeed, even from this, which is from plastic embedded electron microscopy, right there you see the D6 helix, we could map that in. So that means we don't need cryo-EM to do this, to map a whole cell. We don't even need the labels if we can fit proteins at this scale. Labels are better. Here's another example of this kind of fitting. So this is another molecule in Parkinson's disease called LRRK2, of which there are 80 mutations that lead to the disease, known from man. It turns out it decorates microtubules. Wasn't known until we did this kind of labeling and mapping. Okay. Hasn't been solved. But the protein, the domains have been solved. So it has a ROC domain, a WD-40 domain. You can take those structures from the PDB, You can make a pseudostructure out of them, even though this molecule we're interested in itself hasn't been solved, and ask if you dimerize it, which is the way it's thought from biochemistry to occur, how does it fit into those side arms that you see from this kind of tomography, and it fits perfectly. So this is one way to get to the molecular scale. I'm just trying to impress you that we can go all the way to the scale and work with the wonderful library of the PDB. During all of these years that we've been doing this, we've been living in this building as well, asking how can we use these advanced visualization techniques to put all this data together? How can we pull things out of the cell center database, now the cell image library, cell center database, and annotate and assemble across scales? If we want to build a cell based on its components that we've mapped, like this mitochondria where we're taking the cristae apart, and we want to work in that environment and we want to do annotation, can we annotate with the right terminology that comes from the subcellular anatomy ontology that Marianne's group has been building? We can do that. We can work in a fully immersed 3D environment. This is in the next hall. There's this thing, I think it's still running, called the Verrier. And this is one of our students, Iman Mostafavi, he dropped out of graduate student to start his own company. He had the second most popular iPhone app. Okay. But he's fully immersed in this cell, pulling things around and exploring. This was great for raising money, but terrible for thinking about how it would scale to the community. My thinking at the time was, yes, I need to raise money, but what will commodity screens look like? Will Samsung make a tablet that's 3D or a phone that's 3D? I predicted correctly. And so we decided immediately to start working on this software environment that would be portable to commodity devices. First with a downloadable application using an open source game engine, and as you'll see later, using now the Google Body Browser as an alternative. So I'm expecting Iman, since he's making a lot of money now, to donate to this project. So I went to a local benefactor, Ted Waite, who also funded the Waite Biophotonics Institute, the Salk. Waite started Gateway Computers and lives next to campus. He asked me, Mark, how can I help you and what would be the most exciting thing to do? And I said, well, let's build the Google Brain down to the cells and the cell as well. And the idea was very simple, that we take something like Google Earth, and the notional equivalent is if there's a Wikipedia, then we use something that's the equivalent of Wikipedia for neuroscience, which Marianne and her colleagues have a contract to build for NIH called NIF or Neurolex. And if the Google Earth was built not only from satellite imagery, but from things that did the street views, let's assume for the moment that the microscopes at the National Center for Microscopy and the community of microscopists that have microscopes in every university are the equivalent of our street view photographers. And we give them an environment where they can drop in what it is they do. While this was originally done for the whole brain, you can imagine that you could do this for the whole cell. And, you know, we got a little popular press. This is the brain the crowd built. You can see we added the eyes and the optic nerve because the NIH funds us actually to work on eye disease. This is the environment which Steve Larson, a graduate student at the time, really took responsibility for herding the cats to build. And you can see this is moving it around in this game engine. Those are slices from light microscopy. They're elements from electron microscopy. My agreement with Ted Waite was that we would also enable simulations. That in other words, from this environment, if you built a cell and you wanted to simulate it with neuron, which Terry talked about, you could do that on supercomputers by activating the computation from this interface. We did that. But the animation of that simulation can be brought in so that someone can find it and play it, or play the YouTube version of it, because this is open on the web. The other thing that we wanted to do was make this a portal to link out to other resources. So, for example, since this is only a 2.5D environment, you're looking in 3D, we had to put up something that allowed you to move a cursor accurately in 3D. as you scale to large and small. So here, a cursor has been put in this area, and then you right-click, and here we're going to the Allen Brain Atlas, and we're going to look at the gene expression. So now it's piping back to your display things from the Allen site, where they mapped the sort of heat maps of the distribution of all genes in the brain. It gives you the gene expression in that area. Okay. So this is the way you can make an interface that takes advantage of other resources if the spatial or semantic or temporal frameworks are used to link out. Okay. Oh, Phil, we also link out to the PDB, KV 1.2. So you can make a model and import them. If the PDB gave us properties that Tom wanted for a simulation, if it rolled up all the information about, the kinetics of the molecule, it would be even more useful. Here's an example of using this environment in a way to publish. So this is with Anna von Ghosh, one of our faculty who's now heading neuroscience drug development at Roche. We hope he comes back. And Scott Wilkie, an MD PhD here, Eric Bershong, who's part of our group. So this is looking at a region of the brain that they think is important in autism, and a particular kind of spine that's really big, and its development. So here what they did is they used knockdown technologies to basically quiesce the expression of cadherin 9 and then look in specific cells what happens as these synapses are trying to develop. This, by the way, I think will be out in the Journal of Neuroscience, or it just came out. I can't keep track. So here you see the model that came. Again, this is using that serial block face imaging that I showed you, and the tracing of the presynaptic and the postsynaptic components in a normal. And then this is the control for the knockdown and the knockdown. So here what you see is that we inserted those immediately in the right place, or as close as we could get to at least the right neuronal type, in the whole brain catalog. Now we used here, I think Tom notices, this is one that was simulated, just because that's what we had in there. And we placed it in the right place, and you can see then, if you want to hunt around, where this reconstruction might live. Could be more accurate if you're doing the brain in general, but I think you get the picture. And so when this article is published, the intent is for people to be able to find this data. In fact, the paper references to the cell-centered database. This is fast-forwarding this environment to what I think you all wanted to see was, could we use the Google Body Browser, the Zygote browser? So this is taking a granule cell and showing you how we peel away, look at the mitochondria, play with different components. So Google spent a lot of money in what was an open project coming up with a very nice interface for interacting with a 3D scene. We figured we don't have that kind of money to do the kind of interface development. Since this was open, we just take it. Of course, that's what was intended to be done. And so this is what we've built our future projects around, is this kind of an interface going all the way from the mouse to visible virtual cells pointing to simulations. Any of you who want to see this, this shows this project for the cell data bank as we envisage it, unfunded. But I won't go through this, it's a 10-minute video. But this is just a movie, which I think Graham has seen but probably appreciates because it's from, I think, an equally talented animator friend, Drew Barry. It shows what we intended the whole brain catalog to look like. Drew got a MacArthur Genius Award, so he's continuing to do this sort of cellular and subcellular animation. But this takes, again, a storyboard that we came up with for how you would take advantage of assembly of visible virtual neurons in their ecosystem and ask questions. So the neurons that you see here are granule cells in the hippocampus. And those are the neurons which, if you're a healthy adult and you exercise, you sleep well, you have a crop of them born when you're trying to learn something. So even though they say you're always losing your neurons and by the time you die you've lost a lot of them, if you're healthy, you're continually giving new neurons a place in your brain. Now, in an animation like this, we can deal with the electrical activity, as Hollywood often does, as flashes of light. So we not only deal with the tyranny of spatial scales, navigating from, you saw the whole mouse, and we'll go down to molecules in a minute. We can deal with the differences in dynamics by light and by sound. So I paid someone to compose the music for this, and we thought a lot about it. How do we communicate temporal scale? So you hear the tempo changing. So at the same time all that electrical activity is going on in milliseconds, in minutes to hours, maybe days, the processes with actin inside of them are extending from these dendrites, looking to hook up and make new synapses. So we thought to do that with music. Maybe not very well, but I think it emphasizes the problem of multiple spatial and temporal scales and how you look at them, how you interact with them. That process is not allowed to hook up unless this yenta, this glial cell, sprinkles fairy dust, the same fairy dust that's involved in wound healing, thrombospondin. So in a way, learning is the brain's response to the injuries of experience. And what was at first cacophonous becomes melodic. And of course, this is happening on a microscale. Those were receptors. But in the body of a single astrocyte connecting the activities of multiple neurons, Marcel knows I have a glial-centric view of what's important. And this is supposed to say funding to be continued, but we're still looking for. So with that, I think I've kept you long enough, and we should drink.